nucleoside consists of nuclear bases, nitrogen bases, or simply bases. And all these words amount to the same thing. The nucleoside also has a five carbon sugars, either ribose or deoxyribose. So it's either ribose or deoxyribose. Whereas a nucleotide is composed of a nuclear base, 5 carbon sugars, and 1 or more phosphates. A pentose sugar is a monosaccharide sugar, and it has 5 carbon atoms. And the combination of a base and a sugar is known as a nucleoside. Right, if I can just bring this over to show you something here. Now, I want to just show you that the A pairs up with the T. This is for the DNA, and the A pairs up with the T. The G pairs up with C. Now, uracil only found in RNA, and it replaces the thymine of DNA, and so that the uracil is paired up with the A there, the adenine. Now, I've got a DNA here because I'm doing the base pairs of AT, GC, TA, AT, CG, GC, and the hydrogen bonds are sort of around that area, and the phosphate and the phosphate sugar backbone, and the sugar varies because obviously it's deoxyribose here, but if it was RNA, it would be just the ribose, and so that's how it works. If you look at the compound C5H10O4, now that one is DNA, but for the RNA, it's got one more atom of oxygen, and the deoxy obviously, hence that's why it's lost an atom and it's gone down to 4 atoms. So they're basically the same apart from the oxygen atom there. So yeah, I just wanted to show you that, and I'll just put that down then, and I want to just mention something else now. Eukaryotic cells, now they're organised into long structures called chromosomes, and eukaryotic organisms store most of their DNA, they store their DNA, inside the cell nucleus as nuclear DNA. Now, and some of it is in the mitochondria. It's known as mitochondrial DNA. In contrast to prokaryotics or prokaryotes, and they're stored, there's DNA which is stored in the cytoplasm as circular chromosomes. Yes, and now plasmids, they're small circular double-stranded DNA molecules, and it's distinct from the chromosomal DNA in bacteria. Because, as we said, the genetic material of prokaryotic circular DNA is single molecule or chromosome. It's in the form of a single molecule or chromosome, and it's double-stranded DNA. And the DNA in prokaryotes is the central area is called a nucleoid, and it's spelt NUCLEOID, and that's basically it. And I just wanted to say one more thing, actually, that the ribose is one oxygen atom attached to each carbon atom. So, and I'm just basically talking about the building blocks of DNA. And just mention a couple more things. The DNA is packaged in chromosomes in a structure called chromatins, and the chief protein in this is a component called histones, and it's spelt HISTONES, and that's and then I'll mention some other things. The DNA molecule is made up of a double chain of nucleotides in the form of a helix, and the nucleotide bases in the helix pair up with AT and CG, yeah, okay, and yeah, that's it.